So today we are going to learn about serotonin. We have to know that serotonin neurons or serotonergic neurons are located in raphe nucleus. Okay, so few things which you have to know is what is the other name of serotonin. The other name of serotonin is this 5-hydroxytryptamine, this 5-HT. No, the first thing that should come into our mind is what is serotonin. Serotonin definitely is a neurotransmitter. Where is the serotonin neurons are located in our brain? They are mainly located in raphe nuclei, which lies very close to the midline throughout the brain stem. So it is present in the raphe nucleus. Now, what is its function? Its main function is that it's a neurotransmitter of happiness. So it is somehow going to modulate the pain, reduce the pain sensation. It is important for sleep as well as for biological clock system. So let us go into detail about its synthesis, about its pathway, how it is inactivated, and what are the serotonin receptors and what is its function. And then we can go about what are the different manifestations if there is an increase in serotonin level or if there is any decrease in serotonin level. So this picture which you are seeing here is actually depicting the synthetic pathway. So what is its substrate? It is serotonin is synthesized from tryptophan. Tryptophan is an amino acid which gets into the circulation. It gets into the neurons, the synthesizing neurons from the circulation, from the ECF via the sodium tryptophan co-transporter. So this co-transporter is going to transport sodium along with tryptophan. So tryptophan is my you can say precursor or the substrate for the synthesis. And this level of tryptophan, that is plasma concentration, can affect my serotonin level. Definitely we are going to depend on to the nutrition for these tryptophan. So how are we going to do? What are we going to do? What is the rate limiting step here? We are going to study about it. So when it comes to synthesis of serotonin, the precursor of serotonin, as we see here, is the tryptophan, and it is an amino acid. The plasma concentration of tryptophan is dependent on our dietary intake. If our dietary intake doesn't have sufficient tryptophan, probably that is going to get reflected with the amount of serotonin that will be present in our body. So let us start with the steps. Tryptophan gets converted to 5-hydroxytryptophan in the presence of a rate limiting enzyme, and this enzyme is what? Tryptophan hydroxylase. Tryptophan hydroxylase is the one which is going to do the conversion of tryptophan to 5-hydroxytryptophan. And since this is a rate limiting step, we call this step to be very important in the sense, if we have more activity here at this step, the amount of serotonin is going to be more. If it is less here, it is going to be less. So definitely the amount of serotonin is going to be dependent on this step of conversion of tryptophan to 5-hydroxytryptophan. Next, it has to go for decarboxylation. So we have got 5-hydroxytryptophan, which has to get converted into 5-hydroxytryptamine. So serotonin, the other name is 5-hydroxytryptamine. Okay, 5-HT we call it. Now this is called for decarboxylation. So what is the decarboxylation step and who is the enzyme present here? For the decarboxylation, the enzyme which is required is L-aromatic or aromatic L-amino acid decarboxylase. Okay, so this is the same enzyme which was present in our catecholinergic neurons, and this is going to get converted into 5-hydroxytryptamine. Serotonin going to go into our synaptic vesicles. So as we know, for most of the neurotransmitter, there is an antipode system where the hormone the neurotransmitter gets into the vesicle, and against to that the hydrogen ion is coming out of the vesicle. So this is called as vesicular membrane transporter. So this is a vesicular monoamine transporter, the same transporter which was present for my catecholamines. They are the same one. So once the serotonin gets into these vesicles, it stays there and it waits for the impulse to come. So suppose we have an impulse, an action potential has reached this neuron. So what is this neuron going to do? This neuron is going to activate same So high firing rates are going to increase the calcium, there are voltage gated calcium ions. This is going to increase. Then what else is going to happen? Once this has increased, this synaptic vesicle is going to go and release the neurotransmitter into the synaptic cleft. Now what is happening now? This neurotransmitter which has come into the synaptic cleft, it has two ways to go, isn't it? One is to act through its autoreceptor, and autoreceptor, the type of receptor which is present here is 5-hydroxytryptamine 1D receptor. So we should know that serotonin receptors, there are many subtypes of serotonin receptors, almost all except for 5-hydroxytryptamine 3 receptors, which is G coupled protein receptors. Most of them, they are going to act via cyclic AMP. Anyways, we are going to come back again with another slide where we might talk about the receptors. So we have an autoreceptor here. Autoreceptor, we already know. What is an autoreceptor? Can anybody tell me? We have talked about autoreceptor even in dopamine also, where we called that D2 is a autoreceptor there. Yes, please answer.
It's a receptor on the presynaptic membrane that controls the amount of neurotransmitter that's released.
So it is present in the presynaptic end, and it is going to cause the regulation of release of its own transmitter. So auto is means self. So there is a 5-hydroxytryptamine 1D receptor present in the presynaptic end, which is going to act like your autoreceptor. Then what is the other fate? This is fate 1. What is the other phase? The other fate for this neurotransmitter is to get transported back, or there is a reuptake of it. Okay, here you have to mind it. There are many antipsychotic drugs, who are going to block this transporter. So 5-hydroxytryptamine along with sodium, with the help of 5-hydroxytryptamine transporter or serotonin transporter, is going to get transported inside the neuron. So once it is transported inside the neuron, what is it going to go for? It has another way. It can go for oxidative deamination with the help of MAO. MAO is monoamino oxidase. And we will find a metabolite, a principal metabolite of serotonin, which we call as 5-hydroxyindole acetaldehyde. Okay. So these are the few names. So very quickly we are going to revise. So let us just go very quickly and try to see the life cycle of it. So who is going to tell me the life cycle of it?
Hello.
Yeah.
So
yes.
First of all, so the substrate that is yes, ma'am. So the substrate is tryptophan, which the main source of is which is dietary. So from tryptophan via the sodium tryptophan co-transport symport system, it will enter the presynaptic axon, but the terminal axon terminal. From there, it will undergo hydroxylation by tryptophan hydroxylase and give rise to 5-hydroxytryptophan. This is known as the rate limiting step because it is the concentration of this enzyme as well as the concentration of tryptophan is going to determine how much serotonin or 5-HT we finally have. So this is the most important step. So after 5-hydroxytryptophan is formed, it will undergo decarboxylation in the presence of enzyme.
So decarboxylation means where carbon dioxide is released.
Yes. Yes, ma'am. So this will be under the enzyme aromatic L-amino acid decarboxylase. As the name suggests, it's a decarboxylation process, which will give rise to 5-HT, which is 5-hydroxytryptamine or serotonin. So from this, of course, we have to transport this into the vesicle, which will happen in the presence of the vesicular monoamine transporter.
So just remember that there is a vesicular transporter here.
Yes. So from this, the vesicular, it will be encircled within the vesicle. From here, it is going to wait, of course, for an action potential, for a strong enough action potential when it is generated. It is going to cause the what is it called?
This one, entry of calcium, voltage gated
calcium. Yes. So based on the ion gated,
there will be depolarization. So hence sodium is also shown here. But what we are mainly focusing on is the entry of calcium ion through the voltage gated calcium ion channels. Okay.
Yes. And then after when enough depolarization takes place and enough calcium ion has entered, finally there will be release from the vesicle of the 5-HT into the synapse. Some of which will, of course, go into the autoreceptors itself, which is the 5-HT1D receptor, which is an autoreceptor which is present on the presynaptic membrane itself, and is going to regulate the release of 5-HT. Some of it is also going to be taken up, go for reuptake via the 5-HT transporter system. Here again, it is going to the same transmitter is going to go back into the same axon terminal. And some of it, of course, will undergo degradation in the presence of MAO, monoamine oxidase.
Let me just tell you one more thing. You know this, right? For others also, this is very important. Why this is very important? Because if we use inhibitors, mainly the antipsychotic drugs or antidepressants, usually are of SSRI. We call them as selective serotonin reuptake inhibitors. And what is their role and how are they going to bring about this action is they're going to block this transporter. When they block this transporter, reuptake of serotonin is kind of inhibited for some time till that drug is blocking this, and there will be increased availability of serotonin in the cleft. So we'll have 5-hydroxytryptamine over here, and that is going to be available for more period of time. So this is how the person will feel better, or maybe it will reduce their symptoms of depression. So usually the antidepressants, if you remember, most of them will come with the trade name of others, but if you come to their activity, they are SSRIs. SSRI stands for selective serotonin reuptake inhibitors. Okay, so then we might have certain stimulants, which is going to compete here for their reuptake, and that is how they are going to somehow be applicable in a state of euphoria. We have seen that such type of drugs which is going to compete with 5-hydroxytryptamine for its reuptake usually are the one which will relate to the feeling of euphoria.
What I perceive euphoria to be, I think it can be described as a state of extreme happiness.
Yes. So when you have more amount of serotonin present in your synaptic cleft, you are kind of being more happy, unnecessarily being a state of high rise, too happy about it. And that is mainly because of certain drugs, some of the psychostimulant drugs. What do? They compete with 5-hydroxytryptamine for the reuptake. So they just don't allow this one to go into, rather they themselves will sit here. And so what will happen to this concentration? It will get raised in the cleft. Hence, there will be a state of euphoria. Now, coming to the metabolite. So, if we start with the first stage. Yes, somebody wants to say something. I'm not into your screen. Let me come back and check.
So, ma'am, could you explain once more why the second stage is known as rate limiting stage? The rate limiting stage.
Rate limiting stage. Okay, let me repeat it again for you. So this is the first step. Okay, you have to understand this is tryptophan. What is this tryptophan? It is an amino acid. Where does it come from? From my dietary intake. So dietary intake in the sense is, suppose this is my GI, okay, and I have my small intestine, then there is a large intestine. So I have taken some dietary intake. There will be digestion, ingestion and all that, and there are blood supply to my GIT. So what is happening? The nutrients is taken into the system. They go to the systemic circulation, and I have tryptophan present into my ECF, in my systemic circulation. Now, this tryptophan has got a receptor here, which is going to bind along with sodium, and there will be what you call, say, a transport of tryptophan inside the serotonergic neuron. See, we are talking about a neuron which is serotonergic, means it is going to synthesize serotonin. Tryptophan gets converted into 5-hydroxytryptophan. This is in a step of conversion. And who is the one who is going to regulate it? Is this enzyme. And this enzyme is called as tryptophan hydroxylase. It is rate limiting. When we say any enzyme is rate limiting, means when the activity of this enzyme is increased, the concentration of your 5-hydroxytryptamine will also be increased. Okay. So probably if I regulate this enzyme, I will be able to regulate how much of tryptophan do I need, whether I need it more, whether I need it less. Okay. Next is the next one. Once it has been hydrolyzed, it needs to be decarboxylated to form my 5-hydroxytryptamine, which in other word is my serotonin. So this enzyme is called as aromatic L-amino acid decarboxylase. So now what has happened? Your 5-hydroxytryptamine is formed. Now this has to go and get stored in the synaptic vesicle. How we have learned in all the other neurotransmitters that there is a uptake of neurotransmitter into the vesicle, and in that who is involved? An antipode system is involved, where serotonin is taken in exchange to hydrogen ion. So this is going to remain happily in the synaptic vesicles, present here, everywhere. They're going to be happy here. Now, suddenly what has happened is, it is going to get stored in the vesicle, remain there, do nothing, until unless it gets an impulse. It gets an impulse via this, some maybe some stimulus, some impulse, it is going to come here. When action potential reaches to the terminal, this is going to cause the depolarization here, isn't it? For depolarization, we want what? Cations to get inside the cell, isn't it? After all, neuron is also a cell. So when cation gets in, we are basically talking about the calcium ions. When we say that there is a change of voltage here, we are referring to the calcium ions. Along with that, some sodium ions also will influx. Now what is happening is, once calcium gets inside, there is depolarization of the presynaptic end. There will be release of neurotransmitter by the process of exocytosis. Am I clear till this point?
Yes.
Now, once the neurotransmitter is released, it has two ways to go. One is that it can just go back to its own receptor. Along with its own receptor, it's going to get, going to stimulate either the activity over here, or it can just go and stay there. It may act through its own autoreceptor in a feedback manner. The other thing is that there can be a reuptake. Reuptake means you're going to take it back as it is. You're going to take it back as it is, and for that we need a transporter. This transporter is 5-hydroxytryptamine transporter, and this is a co-transporter or symporter, where along with the 5-hydroxytryptamine, sodium also gets inside. Once this 5-hydroxytryptamine is there, again it has a two fate. One, it can go back and stay here in the synaptic vesicles. It may be taken back in the synaptic vesicles, get stored there, or it can be metabolized. And for its metabolism, we require an enzyme which we call as MAO. MAO is monoamine oxidase. And this monoamine oxidase, what is it going to do? It is going to metabolite my 5-hydroxytryptamine into a substrate or into a compound which we call as 5-hydroxyinidole acetaldehyde. Okay, so this is a principal metabolite of serotonin. Now here, what is the clinical application? The drugs which are used, because we know serotonin is a hormone of happiness. So we want to, if somebody is in depression, so what do you think is a condition of his serotonin release? Will he have it more or will he have it less? Less. Okay. Yes. So if he has less, definitely less will be active, means less will be available for its action, and the person will be in a state of depression. Now the person has got a chronic state of depression. See, depression is something which we all face one or the other time. Okay, it's not that every time that we are depressed is a clinical type of depression. Once or the other time, it may be that due to a mood change, or maybe due to some after effect of some thoughts, or maybe due to weather. Sometimes we feel depressed if it is very gloomy, no bright sun, isn't it? There have been situations like that where we feel too depressed at that time. So definitely our serotonin hormone. Yes, somebody wants to say something?
Ma'am, I just wanted to ask if MAO was activated like highly by any mechanism, then also it could lead to depression, right?
Yes, that also it could lead to depression. You're fine. You're right about it.
And ma'am, even the symporter, if the tryptophan and a sodium symporter, if there is a problem there itself, that could also lead to depression. Right?
Yes, yes. See, there are many reasons. Anywhere, any problem in the pathway. So that way, Harshita, I can say if I don't have a proper diet, I'll have a depression.
Yes, ma'am. Because
starting itself. Yeah, I will not have my substrate for my synthesis of serotonin, right? Yes, but usually, the quantity that we require as a neurotransmitter is not in a huge amount. We hardly need few amount of it, you know. So it's just that we usually don't face face this issue. Most of our diet is meeting up and giving us the tryptophan. But yes, malnutrition will lead to depression. That is one major thing, that maybe person who is malnourished, you know, will have depression. He's not going to be very happy. Have you seen any Somali kids being very happy when you go and click their photos? Isn't it? We have seen in internets so many malnutrition kids, we have seen their pictures. I don't think looking at their facial expression we ever feel that they're very happy, right? So might be this is one relationship which we can draw in terms of neuroscience. When I'm talking about, in terms of that, we can relate that yes, probably we don't have enough of substrates. There is no serotonin getting released. So see, where all can be the problem? The problem can be initial stage of synthesis. There can be an issue of reuptake, you know, this issue. It can be an issue over here, okay, or it can be an issue over here also. I have more of this MAO is overactive and it's just changing, just degrading all of my, breaking away the 5-hydroxytryptamine into its metabolite, principal metabolite. Metabolism is very high. Okay. Now let us come to the receptors. As I told you, serotonin, there are many subtypes of receptors. So as you can see here, let me just take the pointer. So as you can see here, there are many subtypes of receptors for serotonin. So what are those? Except, let's just write it, except for 5-hydroxytryptamine, except for this, except for this person who is 5-hydroxytryptamine 3. Okay, except this receptors, all are actually what they are, GPCRs. Yes, we also had one discussion about it. Who all has prepared about the G protein coupled receptors? If I say GPCR, it is G coupled receptor proteins, or G protein coupled receptors, you can say. Okay, G protein coupled receptors. How many of you have tried writing it? We are almost coming to an end of this week. And I think the starting of it only we had tried to, last week we discussed about it, right? Where I had asked you to write about this G protein. Has anybody written anything? And otherwise those people who have submitted at least can discuss over here. I know there have been few submissions. Can anybody discuss about it? When I say G protein coupled receptors, I have G proteins. No? No one has done it? Okay. So let me explain. Let me wait for you to come up. There had been few submissions where some people have tried to submit it. So what we say here, for 5-hydroxytryptamine receptors, when we are talking of these receptors, so I'll wait for you to read up and come. Otherwise, I would have now had a discussion with you regarding that. I thought this was a good time to discuss. But since you are still not ready, let us wait. But tomorrow, definitely with all positive notes, everybody is going to read up and come and tell me about G protein and how is it going to act in my intracellular communication. Okay. So as I say, except for this, all are G coupled protein receptors. So this another thing what we have to take care is this one. The protein 5-hydroxytryptamine 1, the receptor 1, is actually having a subtypes like A, B, D, and for 5-hydroxytryptamine 2, we have another subtypes like 2A, 2B, and 2C. So what are these subtypes going to do? So 5-hydroxytryptamine 1, H1 subtypes, they are going to decrease the cyclic AMP. This one, these subtypes, what are they going to do? They are going to decrease the cyclic AMP. This is one thing you should know. While these ones, TH4 to TH7, these are going to increase my cyclic AMP. Okay. So what happened? The 5-hydroxy, now how about this protein? 5-hydroxytryptamine H2 subtypes. They are also coupled via the G protein and the phospholipase C. So they are excitatory in nature. Okay. So this is just for your information, not much to remember about it. But one thing you can say that these 5-hydroxy H3 are ligand gated ion channels. This is actually a ligand gated ion channel. So it is going to act similar how it is with the acetylcholine, where acetylcholine comes and binds it, and then that same receptor where the acetylcholine comes and binds acts as an ion channel. So 5-hydroxytryptamine 3, this receptor is going to act like how my nicotinic receptors are, and they are also pentamers and they act like an ion channels. They also have many isoforms. We are not going to go in much detail about it. But why have I brought this into your knowledge is, this is one receptor whom we will try to block or use an antagonist. We would like to block it by using an antagonist in case of, this is the story for the drugs who are going to act as antiemetics. What is this antiemetics? Anybody had heard this term called as ondansetron? Have you heard of this drug, ondansetron, which we use for people who are having vomiting or nausea? You know, this is a drug which is usually given when a person is complaining of vomiting or complaining of nausea. Mostly it is given when vomiting. Now, in kids, we don't give ondansetron. Instead of that, we give, Kriti, you can tell, we give Domstal, right?
Yes, ma'am.
We don't use ondansetron. Yeah. So now, what are these antiemetic going to do? Antiemetic drugs, they actually block this receptor, 5-hydroxytryptamine 3 receptor. So we have 1, 2, 3, 4, 5, 6 and 7. This third one, why am I being talking so particular about it, is because the drugs who are going to stop your vomiting, because this is one receptor, if you block this, and where is this area? The area where these are more present, and the one which is responsible for your vomiting is called as vomiting center. We call it as vomiting center. And if you want to know the anatomical, we call it as area. Okay, we call it as area post tremor. So I will try to summarize what all we have learned till now. How are we going to summarize about it? First of all, we should know that serotonin receptors, they are in many isoforms. Let me just take my point. 5-hydroxytryptamine or the serotonin receptors, they are present as many isoforms, 1, 2, 3, 4, 5, 6, 7. Except for this one, the third one, all others are going to act via G protein coupled receptors. So they are going to activate the G protein system. 5-hydroxy 1, this transporter receptor 1, is having again the isoforms as A, B and D. 2 is also having isoforms. What is their function? They usually decrease the cyclic AMP. And how about the rest one of them? They are going to increase the cyclic AMP. But the point to remember here is this receptor, that is receptor number 3, 5-hydroxytryptamine 3. It is very important because there is 2 features that we have to remember. One, that it acts like a ligand-gated ion channels, very similar to our acetylcholine receptors. And the other most important one is that this receptor, which is highly present in the vomiting center, that is area postrema, is going to get inhibited if we use antiemetic drugs like ondansetron. Okay, so they are going to block those receptors which are present in the area postrema. So the area postrema is actually a chemosensitive cells which is going to trigger your vomiting in response to any toxic substance which is there in the blood. This is vomiting itself is a big reflex. We are not going in detail about it. So this is about the receptors of serotonin. Have everybody understood this? It's a small way of telling, we didn't go into detail about it, that where are these presents, how are they going to act, what are the antagonists for this and agonists for it. You should just know that most of their agonists are used as anti-anxiety drugs. So mainly when we are talking of antidepressants or anti-anxiety drugs, we are kind of looking at 2 ways of treatment. There are two ways of treatment. What is the two way of treatment? How am I know? One is that either you are going to increase the concentration of the hormone, concentration of the neurotransmitter. You are going to increase the concentration of the neurotransmitter. Where? At the synaptic cleft. Then you are going to decrease the reuptake. So this is the principle of treatment. Either you decrease their reuptake or you decrease their degradation. So you are going to decrease their degradation so that they are present here for long period of time. Okay, or you can have somebody who is going to mimic your the activity of serotonin, somebody synthetic way who is going to mimic your neurotransmitter which you actually want in your who is of your interest. Okay, have everybody understood this?
Yes.
Yes.
Okay, it's quite clear, not so difficult to understand. Now, let us go here. Now, this is a big list of it. You know, where they are present, what do they do, who all they are. So, I have already told some of them. 5-hydroxytryptamine 1A. See, most of them are going to decrease the cyclic AMP. Did you see that? 1, 1B, 1D, 1E, 1F. All of them are going to act through G inhibitor, G protein inhibitor, GI, and they are going to decrease the cyclic AMP. Then the rest of them, I'm not talking about this. Can anybody tell, see this? This is totally different. The one which is acting for my, yes, somebody wants to say something? Now, let us come back to where we were in our study. What are we trying to tell here is the receptors. Did you all get it? Is Shaili, your question clear to me? Because that the whole topic is totally going to take up our time and will be lost from our track of today's topic. Okay?
Yes, ma'am, I understood.
Okay. So now as we were talking about, as we were all talking about these receptors, what is the thing that we have to keep in mind? That these receptors, type 1 and its isoforms are going to decrease my cyclic AMP. Then the other ones from 4 to 7, they are going to increase my cyclic AMP. These are actually, other than these 5-HT3, almost all are actually G protein coupled receptors. So they are going to act either with the stimulatory GS, G coupled protein stimulatory GS, or G coupled protein inhibitor or GI. Okay, or they may act with GQ also. But what is that which we were trying to focus is this, that the third receptor, 5-hydroxytryptamine 3, they are actually ion channels. And they are very similar to our acetylcholine receptors. Once they are open, once the ligand comes and binds to them, they are going to be open. What is the other clinical aspect we have to remember is that most of the anti-emetic drugs are going to block this channel so that they can prevent you from vomiting. Now look at the effects and look at the distribution. Maximum of them, these receptors are present here in raphe nucleus and hippocampus. So looking at where they are distributed, I probably can tell you what their effects will be. So if they are present with the hippocampus, they are going to act onto your memory. They are going to act along with yours. Now they are with raphe nucleus in the brainstem. So they are going to affect your sleep. They are going to affect your feeding and anxiety. If they are with substantia nigra, globus pallidus and basal ganglia, they definitely are going to affect your initiation of movement, neural initiation, or there will be some inhibition to initiate your movement. There will be behavioral changes, your facial expressions may change. Then it may go to the cortex and hippocampus. I told you there is memory related with this. If it is with globus pallidus and putamen, may cause anxiety again, because basal ganglia along with motor cortex is going to act on, along with other systems, is going to act on to your behavior of anxiety. May talk with, they may be having in the stomach where they are going to affect to your appetite. They may be with the smooth muscles and cortex, may affect your GI motilities, and with obviously not being very specific to the brains, but since all of these areas we have seen. So in total, what do you come to know? That serotonin has got a role and effect to our sleep, to our behavior, to our mood, to our memory, to our cognition, to our learning, to our sleep, to our anxiety, to our vomiting centers, to our appetite center, to our gut motility. So what do you think? Definitely serotonin, since receptors are widely distributed in so many areas of the brain along with smooth muscles, don't you feel that serotonin has got a wide variety of activity other than the CNS activity? Central nervous activity means maybe the cognitions, learning, sleep, mood, memory, anxiety, feeding. It also is going to affect your appetite, vomiting, GI motility. So it has got a role onto the gut also. Now, what are the best foods which is going to help you in boost? It's going to act like boosters for best of the serotonin foods are these. Have healthy diet food. This is what is advisable for everybody. So what is this? These are some pictures which you may find. You must have heard about this a lot, right? Eat dark chocolate in depression. Has anybody of you experienced this also?
Yes, ma'am.
What do you feel? Whenever you are depressed, you take a big bar of chocolate and eat it up, or you give it to somebody happily?
Eat it up, ma'am.
You don't happily share with anyone? Maybe that will give you more happiness next time.
That will give more sadness.
Yes, sharing. Sometimes they say, right, sharing and caring. I don't know why these kids never try to follow that rule. And so much goes from the parents, sharing, caring. But I don't find my kid doing that. They are like, no, it's my chocolate, it's hers chocolate. She has to eat hers one. And if she's not eating it, it's me who has to eat hers one also. So anyway, this is about some of the foods which I've downloaded from the internet. You are better readers there. You can go and try to explore as what you should eat to remain very happy. Now, for me, what is important is the pathways. Okay. So let us talk about the serotonergic pathways. Where are they present? What do they do? So actually the cluster of serotonergic neurons are scattered throughout the brain. Look at it everywhere, but most of them are towards the Raphe nucleus. See, this is the Raphe nucleus. Throughout the brainstem, we see this Raphe nucleus being present. So that's why I told you, the moment you hear of serotonin, there are a few words which should come to your mind. One is, it is the hormone of happiness, pleasure. Then, what is it? It is the Raphe nucleus, which is concentrated, most amount of the neurotransmitters and the receptors will find is concentrated around the Raphe nucleus. It has variety of receptors. It's like many isoforms of receptors are present. Except for FS 5-hydroxytryptamine 3 receptor, all of them are G protein coupled receptors. With that basic knowledge, let us go and try to find out where they are projected. So we have projections to the spinal cord. So we have one. Maybe we can try writing here. If you are writing, you can write it along with me. So I'll try to write it for you. So maybe, okay, fine. So maybe they are present where? One thing, they give projections to the spinal cord. Where is this? These ones. They are going to the spinal cord, which may terminate to where? Here is where you have to understand that these fibers which are coming down from the Raphe nucleus. Do you remember the analgesic pathway? Have I taught you? Did I ever tell you about analgesic pathway? In descending analgesics, I'll just give you an overview of that. What is it? See, if there is a pain here, let me just draw a flame. Okay, and you put your finger around here. What do you feel? What will be the sensation that you will get here? Burning. That will be pleasurable or painful? So these pain fibers, they are going to come through the this dorsal root ganglion. They'll go to the substantia gelatinosa, we call it in the spinal cord. And from here, they will go from here and they will go to the cortex. And they are going to give you an information, oh my God, we got a pain here. Imagine of a bullet injury of a soldier in a war. What is happening? That person gets the pain because of an injury and still he is continuing to fight. What is that which is inhibiting him from getting this sensation of pain? And that is what we call as the descending analgesic pathway. Okay. So in descending analgesic pathway, Raphe nucleus forms a very important nuclei. Okay. And this descending analgesic pathway, what is it going to do? It is going to reduce the sensations of the they are going to release certain opioids or morphine-like substances, which is going to block. So if I show here, if this is the pain pathway, okay, here there is a substance P which is related. So there is something called as substance P, which will be released from the sensory input, which is bringing the information about pain. So this fiber is carrying the sensation of pain, and it is firing at a very high rate, and you are getting the pain. You're supposed to get the pain. What if I have somebody coming from the up, and this up person via the intermediary neuron is going to reduce the sensation of pain from here? And how do they do that? They do with the help of a morphine-like substances or endogenous enkephalins. Morphine-like substances, you understand? They are of opioid-like drugs, opioid-like substances, who are going to reduce the inputs, who's going to reduce your action potential, which was firing at a very high rate, like this, it was running. They're going to reduce this rate of impulses, and you feel less of pain. Did you understand this overview of mine?
Yes, ma'am.
Yes.
Okay, do this.
Ma'am,
Yes.
does this pathway and the motivation pathway that you mentioned, do those mechanisms also work when you're exercising?
Exercising, yes, it comes. It comes.
It also can be painful, but you also enjoy.
Yes, yes. It is all see, that way even the sexual behavior is like that. There is a pain, but still there is a pleasure. So there are such situations where you do find the pain, but there is a pleasure associated along with that. That's totally a different way. So let us not get detract or derailed from where we were talking about. So what we were talking about is the serotonergic pathway. What was important for me to tell you here is this. What is that? The Raphe nucleus. I wanted to mention the Raphe nucleus in context to the descending analgesic pathway. So the projections from here, which is going to the spinal cord, they terminate on the nociceptive input. So we have seen how it is terminating on to the nociception is pain perception. Okay, nociceptor is a receptor for pain pathway. To the subthalamic tract, so we have seen how it is going to the subthalamic tract. So let me say here. So can you see that here? So this is a nociceptive pathway. The fibers are going to the thalamus and thalamus to the cortex. So this fiber, since it is going from the spinal cord to the thalamus, we call it as spinothalamic pathway. Is it clear now? So these fibers, who are coming from the Raphe nucleus to the nociceptive fibers in the spinal cord, they are going to somehow reduce the pain sensation. So somehow they are going to reduce the pain sensation. That is one involvement of the serotonin. So no, of the Raphe nucleus, sorry, not of the serotonin, but of the Raphe nucleus. This is what I wanted to tell you. Next, the Raphe nucleus is going to form a circuit along with it, and few projections are going to run to the medial forebrain, the medial forebrain bundle. So this is your median forebrain bundle. It's going to run to that. It may go to the thalamus, somewhere here down to the thalamus. There is thalamus and there is hypothalamus. So it goes to the hypothalamus, amygdala, striatum, hippocampus and neocortex. So hippocampus is here. This is the caudate nucleus. Then this forms your areas here. Here somewhere is your hippocampal area. It goes to the neocortex. So if you see these projections from the Raphe nucleus, the serotonergic neurons, which are found in and around the midline of the Raphe nucleus, this is the midline of the Raphe nucleus, which is present in the brainstem, are involved in attention and other complex cognitive functions. So what are those functions? Since they are involved with hippocampus, they are involved with memory. If they are involved with hypothalamus, they are involved with your gut motility. They are involved with your feeding and satiety centers. They are involved with the neocortex, this frontal cortex. So they probably have some role to play with your attention, with your motivation. Got my point? So these connections are the one for which, bestowed on which you are having the functionality of these neurotransmitters in different areas of the brain. So this is one thing. Some of these serotonergic neurons may also be involved in expression of anxiety, the normal anxiety. So if you inhibit these Gabaergic neurons, enhancing this activity may reduce your anxiety. So now the drugs which are given in anxiety, anti-anxiety drugs, one is they can be SSRIs. SSRIs is selective serotonin reuptake inhibitors, and we have seen where it is acting. It is going to inhibit the reuptake of serotonin so that serotonin remains in the cleft for longer period of time. Then there may be something called as pre-GABA or pre-GABA, which is called as Gabaergic neurons. You know, they are actually drugs who are going to enhance the activity of Gabaergic neurons, which is going to inhibit the activity of other neurons, because GABA is a inhibitory it is an inhibitory neurotransmitter. So what is happening is, so definitely there will be regulation of some sort of serotonin release can be regulated. So here are the certain names. Prozac is the one, Lexapro is the one which are usually given as SSRIs. These are the names here. But one thing you have to also know is that serotonin transmission is modulated by social status. You know, how my position is in my group. So it has been seen in monkeys especially. If the monkey is the head of the group, you know, if he is head of the group, he has a different way of expression of this serotonin. Means his serotonin transmission is actually modulated by his social status because he is the head of that group. So in case of humans, definitely if you have this leadership quality and if you are a leader of your group, of your area, or of any activity which you do, definitely you are going to modulate the serotonin level. Probably you will have more amount of it and you feel happy. Don't you feel that the leaders usually sometimes have this sense of achievement, dominancy? You know, they feel as if, oh, I'm the one who's going to do this and that. So this is how they, you know, this actually reflects your social status.
Okay.
If you are leader to a group, definitely you will have some modulation to the serotonin release. And this has been proved with the animals. Maybe it is applicable to humans also. We never know. But in animals, they have seen that animals, especially monkey being a social animal, it has a group, and the monkeys who are dominant in their group have a different modulation of serotonin. So probably they release more amount as compared to the other ones. And that is why in personality development, leadership quality plays a very important role, isn't it? Don't you think so?
Absolutely. Yeah.
Yes. So maybe this is what the neural regulations is going to have it. But yes, let us now move. Is this slide clear to everyone? Okay. Now let us go to that situation where we have deficiency of serotonin, what we are going to do. If there is excess of it, what we are going to do. So anything which is very high, yes, if you have deficiency of serotonin, you are going to get associated, first of all, you're going to get associated with a symptom of depression. And that may lead to a risk of high suicide. This is what has been seen. So there may be OCD-like symptoms. You know what OCD is? Obsessive compulsive disorder. So here the person does the same thing again and again. You know, if he is tried, suppose he has locked the door and he'll try to go and take the lock, you know, try to hang the lock all the time, check it whether it's locked or not locked. He may just go for washing the hands, means he's continuously washing hands for other things. So there may be obsessive thoughts. There may be compulsive behaviors. There may be suicide and aggregations. Maybe such people, you know, they go for a state of depression. And you now can, I think, relate yourself that when somehow if they are misled and they go towards the drugs, why they feel happier and they try to get addicted. Addiction itself is a different phenomena. I don't have it here, but there is a center which is addictive centers, and de-addiction drugs act on those addictive centers. But here my mentioning is to say that, you know, people who are depressed, if not given a proper counseling and treatment, may either go two ways. Either they will have the suicidal thoughts and they may end up ending themselves, or they may go towards the drugs. Why drugs? Because this drug is going to somehow act onto the modulation. This drug probably has some role in the modulation of serotonin. So it is going to reduce the sense of pain that they have. Okay, maybe it is going to somehow, compete with serotonin for the receptors. You know, they may act like agonist of that, and so they are going to make you feel euphoric. So this is how it goes. And what is one thing we have always seen is that depression in most of the people is linked with food craving. Have you all experienced any time? I don't want the clinical answer, but have you ever experienced this?
Yes.
Anyone? You start feeling too hungry, you know, when you're too depressed. Some people don't eat at all, but most of us usually get for craving, and chocolate is first craving you usually go for, isn't it? So it has got an intersection, you know, the behavior of serotonin deficit and dopamine deficit has got an intersection here at this level of depression and craving. So we have learned about dopamine before, and now we are learning about the serotonin. Now we'll go back here again, how we try to learn up in tables, that if there is excess of serotonin, what is we going to do? It will be called as serotonin syndrome. And what are we going to have the symptoms as? Shivering, diarrhea, motility has increased, muscle rigidity, because again the firing has increased. So you will find that muscles, both flexors and extensors, both have become hypertonic, and now you are giving a sign of rigidity. There may be fever, seizures, agitations, and all. These are the few symptoms. Coming to insufficiency of it. So we know if there is no serotonin, there will be depression. That is one thing we know. There will be anxiety. We know that. Pain sensitive. These people become hypersensitive to pain. Even a small cut, they will have a super pain. You know, like the tolerance level of pain will reduce, because now there is nobody to regulate that pain. Poor sleep, because we found that there is a connection of serotonin with our RAS, with our medial forebrain. So there will be difficulty in concentration, since it is involved with hippocampus and the other structures of the neocortex. Carb craving. Carb craving is for carbohydrate. You feel so hungry all the time, so you look for food at this time, and usually we try to take more of carbohydrate at this time. So there is carb carving, craving they call it. So these are the symptoms that we get if there is insufficiency of serotonin.